very good afternoon to Prof and all my friends. We are from Group 3 and today we will be presenting about quality evaluation and safety of commercially available nutraceutical and formulated products. This Group 3 consists of 3 people, which is me, Aina, Hanif and Raida. First of all, I will start with the introduction. For the introduction, nowadays, the range of dietary supplements that are available for the consumers are increasing and nutraceuticals are expanding sector of this market. Nutraceutical also being studied for the development of possible medicine for major disease treatment including cancer and Alzheimer's disease, and the use in diet supplementation for prophylaxis against a number of diseases. In 1989, the nutraceutical is defined as food or parts of a food that provide medical or health benefits and the prevention and treatment of disease by Stephen D. Phillips. Furthermore, there are some of nutraceuticals that is not a dietary components, which are generally sourced from plants and plant products such as soy and its isoflavones. There is also naturally occurs in human body and often found in foods while other are animal origin. To summarize, commercial sources of nutraceuticals can be divided into two groups, which are natural sources or manufactured in a biotechnological process. Next, for the contents of single components in formulated products, most of the formulated nutraceutical supplements can be found in widely varying label range of contents such as the resveratrol content that might be claimed to be present in the range of 10 mg to 500 mg in different products. Then, many single constituent nutraceuticals have shown to be simple, non-fraction extract containing a complex constituent profile and contain up to 70% of non-specified constituent. Standard good manufacturing practice, which is GMP of pharmaceutical required 95% until 105% of claim constituent level present. Based on two main references, they are shown the list of monograph of nutraceuticals. The first one is the Handbook of Analytical Methods for Dietary Supplements, which have 5 nutraceuticals, which are chondroitin, coenzyme Q10, glucosamine, melatonin, and soy. For the United States Pharmacopoeia National Formulary, it have 7 nutraceuticals, which are chondroitin, coenzyme Q10, glucosamine, lipoic acid, lutein, lycopene, and maritime pine extract. The most popular products are chondroitin and glucosamine. As you can see, this is table 4.2, which is summary of the therapeutic application, representative chemical structure and dosage levels for the major nutraceuticals. For acetylchemitin, the dosage range is in between 1.5 to 3 grams per day. The sources is from brain, liver, and kidney. Next, for bilberry, it's sourced from Vaccinium methylis, and the dosage range is 120 mg per day. Next, for chondroitin is from bovine trachea cartilage, and it has the range of 1,200 until 1,500 mg per day. For the coenzyme Q10, it is widely range of foods, and the dosage is around 100 to 360 mg per day. Next, for the creatine, it's from skeletal muscle, and the dosage range is between 5 to 25 grams per day. Next, the famous products The next famous product, which is glucosamine. Glucosamine is from bovine trachea shellfish, and the dosage range is in 1,500 milligram per day. Next, for the melatonin, melatonin is from bovine pineal glands, and it can be said that the dosage range is the lowest, which is 0.3 to 25 milligram. Okay, then I will pass to Hanif.
Okay, maybe next slide, Naina. Okay, the next is pinogenol. The dosage is 25 until 200 milligram per day, and resveratrol, the dosage is 15 until 200 milligram per day, and adenosylmethionine, the dosage is 200 until 1,200 milligram per day. The next is soy isoflavones. The dosage range is 30 until 50 milligram per day, and the extract catein is 50 until 100 milligram per day. This is the table about the proportion of product in accepted quality standard from a range of nutraceutical supplements. This is contain the nutraceutical and origin, range of content, and the reference. Maybe next. Next. Okay. This is the contents of single components in formulated product. First is the discrete compositional analysis of 12 Japanese chondroitin supplements sold to products false labelly as being from shark as opposed to their actual bovine origin. Another study revealed that commercially available chondroitin may actually consist of hyaluronic acid. Commercial carnitine has also been the subject of a number of investigations. Carnitine samples show good compliance into surveys, but not into further reports. More recently, two Spanish reports have shown varying quality. And creatine product conformed poorly as did the DHEA and 3-PUVAS and linolenic acid, lutein, and zeaxanthin, lycopene, beta-carotene, and alpha-tocopherol. Complex materials such as soy isoflavones and the extract were also found to have poor compliance. This is the table that show the level of state content achieved by this product were widely variable. There have clearly been fault in the key C or key A department. This product were purchased from a wide range of sources. So from the consumer viewpoint, looking at a range of commercial nutraceutical shows that there is a marked difference in prices. The cost of a number of range have shown wide variation as in table below. More importantly, unit cost of 50 milligram isoflavonoid vary by a factor of 22 from US 0.1 until US 2.19. Next is about content of active constituent of range of nutraceutical of complex composition. Data has been published for a number of examples, including bilberry, branch chain amino acid, conjugated linoleic acid, flaxseed, grapeseed, proanthocyanidin extract, green tea, soy isoflavones, pycnogenol and resveratrol preparations. Published data on constituent level has been driven both by the desire of biologists and clinicians to ascertain precise data concerning the biological or therapeutic activity of nutraceutical supplementation, and also by analysts with an interest developing new analytical method to investigate the ability and application of developing technology. Okay, this is about bilberry. The anthocyanin or glycols of bilberry extract at the same time determine the level of anthocyanate or aglycons that are marker of poor quality products. The latter class of compounds being present due to hydrolysis cause either the storage, extraction, or manufacture. Two products were found to be composed of constituents from berries other than bilberry. There was significant interbatch variation, and one product had no anthocyanin. Labeling error, including botanical species, plant part, and some instances, there was no presence of purported active constituent indicated. And worker using the United States Pharmacopoeia methods for the quantification of anthocyanin contents of 10 commercially available products, not extreme variation in level. Anthocyanin values range from 0 to 44% and anthocyanin from 0 to 6.5%. And the next about branched chain amino acid. And this is more favorable results were found from the analysis of BCAA products of Italian origin, where there were high level of conformity with labeled claims. The next is conjugated linoleic acid. Individual and total CL isomer level in 4US formulated supplements have been found to vary widely in both physical characteristics, color, and solubility, and total fixed oil and level of individual CLE isomers. This is most likely due to the variability of fixed oil source and the condition of isomerization. And next about the flaxseed. Variability in the composition of three flaxseed cultivar have been shown to have different level of activity in a number of biomarkers for atherosclerosis and mental stress, demonstrating the importance of phytochemical composition of biological activity. In one survey of breads fortified with flaxseed, 13 were labeled to contain 0 until 10% of flaxseed. An analysis of latent aglycons resulting from fermentation revealed a wide range of level of the metabolites. Total level range from 1 to 32 micromole per 100 gram in bread, with the metabolite EL appearing as the major constituent in a sample. Similar investigation of 6 breakfast cereal revealed a metabolite level ranging from 2 until 48 micromole per 100 gram, with EL usually present as the predominant metabolite. This result clearly demonstrates no clear link between label claims and actual level of flax constituents. Okay, the next is about grapeseed. A total of 16 Spanish grapeseed products have been studied and shown to have widely differing antioxidant activity and level of active constituent, gallic acid, cyanidins, with a factor of 12x different level. A further 10 products derived from grape skin were evaluated, and detailed quantitative data was shown for level of 13 cyanidins, grape pomace, and its residues from winemaking and juice pressing were also found to contain the cyanidins, and it is likely that these fractions are used in commercial manufacturing of nutraceutical. Maybe Rael will continue.
Okay, thank you for your time, Hanif. Okay, I will continue the presentation from our group. So the next example is green tea. So the green tea levels of claimed active compounds depend upon the method of preparation of the infusion, as well as the choice of tea type. The surface of teas and their preparations have been reported to exhibit wide differences in the levels of catechin derivatives and also theanine. So, the catechins have been shown to be surprisingly stable during normal tea making procedures, but the long-term storage in aqueous media at extreme pH may cause greater degradation as in previously prepared commercial product. The autoclaving at 120 degrees Celsius for 20 minutes was found to cause epimerization of EGCG to minus gallocitazine gallate or GCG, which was found in a number of commercial tea drinks. It probably caused by high temperature treatment, but the stability of GCG itself is liable to further degradation. It's depending on the medium in which it is present, irrespective of a low pH. And then the caffeine levels were higher in teas than the commercial bottle product, which contain minuscule levels, but even lower levels of polyphenols. Generally, fresh leaves contain higher levels of polyphenols than commercial black tea. It is also found from Bonoli et al. Much lower levels of polyphenols in canned green tea drinks than in freshly made infusions. So the next is pycnogenol. So the pycnogenol is the proprietary product manufactured by one individual manufacturer. The variations in constituent levels and proportions of components have been found to exist in 12 commercially available products, but no assessment of the proportion of claimed levels was stated. In addition, one formulated drug was found to contain extra ingredients of citrus origin. The next is resveratrol. The content of resveratrol in grape products has been widely known. White grape juice is known to contain lower levels than that from red grapes, and amounts differ widely with different varieties of vines. In addition to agricultural factors, the resveratrol content of grapes is determined by the infection of the grapes by the fungus Botrytis cinerea prior to harvesting. This results in higher resveratrol content, but this procedure cannot be standardized. As a result, manufacturers enrich the content in formulated products by the addition of resveratrol obtained from the Japanese knotweed, Polygonum cuspidatum. Okay, next maybe. Okay, the next is last soy isoflavones. So the levels of individual isoflavonoid glycosides and aglycons have been reported for soy milks, tofu, and fermented soy foods, including soy sauce, miso, and tempeh, and meat analogue or hamburger products, and wide variations were evident. Variability in levels in soy-based infant formulas available in the United States, for example, has been reported with contents of isoflavones ranging from 214 to 285 micro microgram divided by gram in dry products and 25 to 30 microgram per milliliter in reconstituted products. It's probably caused by the use of different amounts of soy isolate in product formulation. In the further work has also shown wide variation in total isoflavone content in commercial products, with up to 200% variation between products and up to 150% variation in follows in a specific brand. The large variations were evident in both isoflavone content and individual isoflavone in their identical food sources using similar processing conditions as was found from the analysis of 85 soy milks. The another interesting finding reported in one analysis of formulated soy products was that in addition to the variability of isoflavone content in the 14 formulations, there was also present up to 6 lignans, including Sesui solaris tyrosine, albeit at much lower levels. Okay, next. So about the next section is bioavailability. Bioavailability itself dependent on a number of factors, including solubility of the raw material. A wide range of pharmaceutically styled formulations of nutraceuticals are commercially available. Tablets and capsules are widespread, but there is increasing use of more novel dosage forms, including soft gels, various controlled release preparations, chewable tablets, liquids, chewing gums, patches, dissolving strips, oral sprays, and fizzing tablets. The parameters affecting bioavailability include the chemical form of the active, for example, glycoside or aglycone, salt form, and then type of formulation and specific from manufacturing details. Pomegranate is recognized as one of the most powerful sources of antioxidants in plants when tested in vitro, but in reality, the extremely low bioavailability in vivo means that the antioxidants are not absorbed. So the absolute bioavailability of coenzyme Q10 is unknown as it is strongly lipophilic and practically insoluble in aqueous solution and consequently has poor bioavailability. A range of products formulated with emulsifying agents and oil-based facials, as well as fully solubilized formulations were studied in an attempt to improve bioavailability. Improved bioavailability was recorded for the oil suspension in soft gelatin capsule when compared to a standard dry formulation. So the next section is other indicators of quality. Manufacturers often supply formations in dosage not usually employed in clinical trials and often lower and therefore, unless consumers are aware of the levels used for the reported effects, they may be taking dosage below the required amount. The standard assay procedures for pharmaceuticals and nutraceuticals include not only the levels of active ingredients, but also a range of further tests to give an indication of overall quality. For examples of this include disintegration, dissolution, weak variation, friability, and hardness for tablet formulations. Maybe next. Okay, the next section is possible contaminants in nutraceuticals. Overall, levels of contaminants reported in isolated formulations are low. The possible presence of dioxins and polychlorinated biphenyls or PCBs in fish oil supplements available in the United Kingdom was investigated by the FSA, and 2 out of 33 branded liquid and capsule products were withdrawn from sale due to levels that could lead to ingestion of twice the tolerable daily intake. Impure creatine supplements have been shown to contain creatinine and metabolite of creatine present in higher concentrations than the precursor, creatine. This may have occurred as a result of improper manufacturing or breakdown of the creatine as cooled another manufacturing byproduct, disandiamide. Creatine sold in liquid forms is believed to be more liable to break down into creatinine within the formulation. 3 commercial melatonin preparations have been analyzed, and a number of contaminants found in levels up to 0.5%. 6 of these contaminants were related to impurities that had been previously discovered in a sample of L-tryptophan associated with an eosinophilia myalgia syndrome. And then because of the terrorism, human supply not only focused in quality control and insurance. The traceability is now standard in medicine supply using a combination of global positioning, bar or ship coding, and hazard critical control point management coupled to nanotechnology markers assays. This development will certainly follow through into foods and nutraceuticals. Okay, the next section is about safety. Selected LD50 fellows of a wide range of nutraceuticals and doses found to be safe in animal studies have been reviewed. Even allowing for species differences, these selected nutraceuticals appear very safe at therapeutic levels. And the next is adverse effects. Nutraceuticals are frequently involved in basic metabolic pathways in the body, and as such are closely involved in the metabolism of other nutrients. Availability of one nutrient may impair or change the action of another in the immune system, as reported for nutrients such as dietary fatty acids and vitamin A. And the Table 46 lists the incidence of a range of adverse effects of a number of nutraceuticals from data collected by the American Association of Poison Control Centers. Maybe you can click. Okay. Okay, about the table 4.6. Not yet. Okay, but the table 4.6. This is the table of 4.6. Maybe, for example, we have nutraceuticals melatonin, and then the symptoms from the nutraceuticals is drowsiness or lethargy, with the incident cases from the data from Palmer et al. 2003. It's 26 cases, and in single ingredients is 22. Maybe um the four people there is also because the other ingredients maybe like melatonin, and because of the HEA maybe they have some drowsiness. And then the example next example is headache, which is the nutraceutical from creatine in incidence cases is like 35 per people. So maybe the next slide. Okay, the next section is drug interactions. The interactions between nutraceuticals and prescribed medicines or even other nutraceuticals are being reported. There's table again, maybe later. The table 4.7 shows some of these interactions that occur with medicines. Wait, I cannot. Okay, table 4.7 shows wide-ranging interactions between nutraceuticals and medicines, and it is therefore important that physicians are aware of the nutraceutical supplementation being taken by patients. And table 4.8 lists examples of reported interactions of nutraceuticals with either other nutraceuticals or foods. And table 4.8 shows perhaps unexpected interactions between a range of nutraceuticals and other food constituents. The situation will also require more detailed investigation in the future as consumption of nutraceuticals is increases yearly. Okay, maybe you can show us the table. Okay, in the left side, it is table 4.7 about the examples of interactions of medicines with a range of nutraceuticals. So the medicines, for example, sodium valproate, and the nutraceuticals induction of chemitine deficiency. And the next example maybe flufosamine, and the nutraceutical is increased levels of serum melatonin. Is about the drugs. So in the next table in the right side, there's table 4.8 about the interactions of nutraceuticals with either other nutraceuticals or foods. For example, lutein nutraceutical. In nutraceutical or food, it is reduction of beta carotene absorption, and then beta carotene nutraceutical in the nutraceutical or food, it can possible interaction with lutein or lycopene, and levels of alpha tocopherol affected. So maybe next slide. So the conclusions of our presentations is nutraceuticals have applications in a number of therapeutic areas, and some of them show comparable efficacy to conventionally prescribed pharmaceuticals. The second is the quality issue includes complexity and purification of raw materials, standardizations of complex products, the presence of contaminants, and varying effects of different formulations, and requires careful consideration. The third is the majority of nutraceuticals are derived from natural materials, but this doesn't mean that they are devoid of adverse effects. And as outlined, a number of these have been reported. The next is compared to what conventional pharmaceuticals and complementary medicines, nutraceuticals show lower incidences of adverse effects and drug interactions, but it is rising with increasing use. And final is clear government regulation is required in order to eradicate quality problems and limit adverse effects and drug interactions. So it is maybe that was from us, the group